Hello students, welcome to Misostudy. In the today's session, we will be discussing about certain CBSE pattern question exercises for the chapter Biotechnology Principle and Processes. We have discussed about the whole chapter, whole sessions you must have seen. Now we will be revising certain questions, and those questions which I have included in today's session, they are important and also they are from previous year CBSE exam. So let's move forward. So the different category of questions I have included, one from a 1 mark question, 3 question from 1 marks, 3 question from 2 marks, and 2 question from 3 marks and 2 question from a 5 marks. So I will tell you how to attempt such type of questions. So let's move with question number 1, which we have to answer according to 1 mark. Name the technique that is used to alter the chemistry of a genetic material, DNA, RNA, to obtain a desired result. Now in this case, we are altering the genetic material, that is either it is a DNA we are altering or the RNA we are altering. And the DNA RNA alteration that can be done by a biotechnology. Basically in the biotechnology one field is there which is called as a genetic engineering. So the correct answer is genetic engineering. So in the genetic engineering, manipulation of various genes, they are done. Those manipulations, sometimes the addition of gene happens, sometimes there is a removal of gene. This is called as a genetic engineering, right? Now, so the first question, correct answer is the genetic engineering. Let's move forward for the question number 2. So for the question number 2. Here the question indicates how can retrovirus can be used efficiently in the biotechnology experiment in spite of them being causing diseases. Now, here one word which is mentioned, that is called as a retrovirus. Retrovirus, in this we have an example of a HIV. HIV is also a retrovirus. They are having a RNA. Basically what they do the process of reverse transcriptase. They have an enzyme present which is called as a reverse transcriptase, which convert this RNA into a DNA, and later on the DNA is incorporated into a human genome. In the biotechnology, what we want, either a desired gene has to be inserted or some specific gene that has to be deleted. That is called as a gene manipulation. That is ultimately mean genetic engineering is. In this, the vector which is used sometimes is a retrovirus. Retroviruses are those viruses that has the ability to infect any of the eukaryotic cells. They can affect a eukaryotic. In AIDS, when I was talking about AIDS, in AIDS we have studied that these retrovirus or HIV, what they do, they used to infect a normal cell. Macrophages they used to infect, and later on they incorporate their own genome. Own genome first of all reverse transcribed and they form a DNA, and that DNA enter into the nucleus and incorporates itself into the whole genome. Later on multiple copies they are formed, and numerous copies they form a coat, a virus coat, and virus they again come out which is ready for the another infection. This is a normal process. In case of the retrovirus, that is HIV is also a retrovirus that is used in the biotechnology, but that is a non-viral retrovirus. So, non-viral retrovirus. This particular retrovirus is used. When we talk about this retrovirus, they have a capability to insert desired gene into host cell. Right? That's why these are used as a vector. So they are used as a vector, vehicle for the transformation process. So hope the second question is clear to you. Now let's move forward with the next question, question number 3. So here the question indicates name the enzyme that are used for isolation of DNA from bacterial and fungal cells of recombinant DNA technology. We have already discussed about this question. This is a quite a direct question. Even at the same within the same line, such question is mentioned, such a like answer is mentioned. So for the basically what we want, we have a bacterial cell. Suppose we have one bacterial cell and second we have is a fungal cell. First of all, what I want that outermost coat, they should be digested. In case of bacteria, the enzyme which is used for such digestion, this is called as a lysozyme. And lysozyme is also present in our tears, right? Whereas for the fungal, chitinase is used so that the outermost coat, they can be digested, so that a particular DNA of interest that can be easily inserted. That is why it is written over here, that is it is used for the recombinant DNA technology. And also when we talk about when we have to isolate a DNA, what we want that first of all the outermost coat, that should be diluted or that should not be present, and that is being digested with the help of, in case of the bacteria we need lysozyme, in case of the fungus we need a chitinase enzyme. Now let us move forward with the 2 marks. Explain the mode of action of a EcoR1. EcoR1 is what? That is called as a restriction endonuclease. It is a first of all restriction enzyme. In the restriction enzyme we have is a 2 category. One is called as a endonuclease and second is called as a exonuclease. Restriction enzymes are those enzymes which make a specific cut at a DNA. This is a restriction enzyme. Now this restriction enzyme is of 2 types. One is called as an endonuclease, second is called as an exonuclease. When we talk about a DNA, if I want that DNA, they should be cut from in between the nucleotide. Basically what they do, they break that sugar phosphate bond. They break sugar phosphate bond. So if I suppose this is a DNA sequence, I want to cut this DNA sequence. If I cut like this from the center, hope you can see, if I cut like this, such type of enzyme which is required is called as a endonuclease. Whereas when I try to cut it from the end, either from a 5' end or from a 3' end, that is called as a exonuclease. So that means cut in between here, cut from ends, right. This is the difference between the endonucleases and exonucleases. Now let us talk about the question. Question indicates we have to explain about the action of EcoR1. EcoR1 is a very good example of an endonuclease, right. Whenever we talk about the cutting of this restriction endonucleases, they require a specific sequence. So, and that specific sequence is called as a palindromic sequences. They require palindromic sequences for making a cut. And those palindromic sequences, if we read from either this direction or this direction, the result is same. Eco R1 they cut at specific sequence which is called as a this is how they used to make a cut. They cut from in between AT and AT. Now one more. When we talk about the mode of action of the E. coli, Eco R1, if we read this particular fragment from this side, CTTAAG, if I read the same from this side, CTTAAG, these are called as a palindromic sequences, right? So if you have to explain about the mode of action of the E. coli, Eco R1, first of all you have to explain that it is a restriction enzyme. Two type of restriction enzymes are there. The first one is endonucleases, second exonucleases. Then you have to explain about the sequence. Now, let's move towards the next question. How is insertional inactivation of enzyme used as a selectable marker to differentiate between a recombinant from a non-recombinant? So, we have a two cells, suppose recombinant and second is called as a non-recombinant. If I want to distinguish among the two type of cells, the recombinant and the non-recombinant, we always go for one procedure that is called as a insertional inactivation. The name itself indicates insertional inactivation. That means something is inserted due to which something is inactivated. This directly means the same. Now, there are two types of cells. One which is having our DNA of interest, which is added with the help of a genetic engineering. One which does not have a gene of interest, though same type of performance, same type of experiment happened on these cells also, but because of a certain reason, they does not have that DNA or a gene of interest. So, two type of cells are there. Now, I have to identify whether they have that gene of which is of my interest or they do not have that gene. This is a question. What I will do, I will follow one procedure which is called as an insertional inactivation. We all know in the cell the beta galactosidase gene is there which codes for enzyme beta galactosidase. Beta galactosidase enzyme is there and definitely that is coded by gene. So, what we used to do when we talk about the recombinant, that means they are having gene of interest. Gene of interest. Normally, cell have this beta galactosidase enzyme. Gene is there that codes for enzyme. When we do this recombination process, means adding of DNA, adding of a gene of interest, we add at a specific sequence and that specific sequence is in between the genes of which code for beta galactosidase. If in between these my gene of interest is added, now tell me one thing, will that gene will be able to express? They will not be able to express. This is called as a insertional inactivation. So, in this case, gene inserted near or in between gene coding for beta galactosidase. Right, due to which beta galactosidase no express, whereas this is a normal cell, no insertion is there. So, in this case beta galactosidase gene present. Is present. Now, can you see enzyme with your naked eye? Will I be able to see them? Whether it is present or not, it is very difficult for me to check out in this cell, yes it is present, in this cell, yes it is not present. I want some visible thing should be present so that I can get to know whether these are the recombinant cells or the not recombinant cells. So, in this case, a chromogenic substrate is added. Chromogenic substrate. In both the cases, these chromogenic substrate will be added. Beta galactosidase, when it is added with a chromogenic substrate, enzyme is there. In this case, enzyme is not present, so no product will be formed. Means no color formation will be there. No color. Right? Whereas in this case, there will be a color formation. This is an actual difference between a recombinant cells and a recombinant cells on the basis of insertional inactivation. Insertion has taken place, but this enzyme is inactivated, no colored products are there. Whereas those colored products which will be there in the my plate, in the recombinant cells, in the different cells which are having a recombinant or non-recombinant, if some color is there, that means it's a non-recombinant cell. If color is there, if color is not there, that means these are the recombinant cells, right? So, this was a question number 5. Let's head towards the next question. Name the source of DNA polymerase used in PCR technique. Mention why it is used. PCR is what? Polymerase chain reaction. And polymerase chain reaction has 3 steps. One is called as a denaturation, that is a polymerase chain reaction. In this we have is 3 steps. One is called as a denaturation. First step. Second is called as a annealing. And the third one is called as a extension. Right? Just imagine a situation when I want to go for a denaturation. PCR is a normal reaction when n number of copy of a specific DNA is made. Now, denaturation is a process by which the 2 strands of the DNA that has to be separated, and denaturation is normally done at a very high temperature. In the denaturation, the temperature which is required, that is minimum 95 degrees Celsius, and that depend upon the CG sequence as well. So, if this much temperature is there and we want to make a copy of it as well, what will happen? The enzyme, if I add enzyme in initially, normally when I want to make a copy of it, I need an enzyme which is called as a DNA polymerase. So, this DNA polymerase will it be able to work at this much high temperature? It will not be able to work because enzyme is there. Enzyme is made up of protein. Protein naturally it will denature. Enzymatic activity will be lost. So, what I want, first one step will happen and the second step will happen. In the third step, I need a DNA polymerase. Those DNA polymerase which can be added in the extension process. In this, again and again I have to add this DNA polymerase. So, this was a drawback of this particular, you can say, instrument, that is a PCR. So, Carey-Mullis, he used one enzyme which is called as a TARC polymerase. TARC polymerase, and that TARC polymerase is obtained from Thermus aquaticus, which live in hot springs. That's why they have a tendency to even sustain at a very high temperature. So this is the correct answer, that is a Taq polymerase is used, which is obtained from the Thermus aquaticus, and the name itself, the name, the source of the DNA polymerase, and mention why it is used, because the temperature during a denaturation step is very high, and also again and again adding the DNA polymerase at the extension time, it's very difficult and it's very time consuming also. That's why the Taq polymerase, which is obtained from this bacteria which lives in a hot spring, they can easily survive with this much high temperature, that is used. So this was a question number 6. Now let us move towards another category of question, that is a seventh question, and that is a question of 3 marks. So question number 7 is of a 3 marks. Question states, explain the role of following in biotechnology. First, restriction endonucleases, second, gel electrophoresis, and third is a selectable markers in PBR 322. First of all, the first we have is restriction endonucleases. Restriction endonucleases. So restriction endonucleases, they are called as a molecular seizures. Why we use this molecular seizure is that we want to make a specific cut in the DNA. That is done with the help of these restriction endonucleases, or called as a molecular seizures. In this you have to explain about the 2 type of ends. One is called as a sticky end. We have explained, we have already done in the session. So sticky ends, and second you have to explain about the blunt ends. What is the significance of these 2 ends? And also you have to explain about the palindromic sequences. Palindromic sequences. And you can write the 2-3 examples of it, like EcoR1, BAMH1, HIND3, PVU, etc. You can write in this. Second part we have is the gel electrophoresis. Gel electrophoresis. So gel electrophoresis is a process of separation of DNA fragments in agarose gel by passing electricity, Students, tell me one thing. When we talk about the passing the electricity, or the separation of the DNA fragment is happening, how the DNA fragments, they are separated? By passing them from electricity. We know electricity, they are having some poles present, a cathode and anode is present in this case. To talk about the charge which is present in the DNA is negatively charged. Like I have explained in the sessions, similarly you have to write here as well. I have made a diagram in the session. Try to make the diagram over here, with which you can easily explain the things, how the DNA fragments, they are separated. DNA is negatively charged, they can easily move towards anode. So they will move towards the anode, and like this there will be a transportation, due to which the DNA fragments, they will be separated. Larger DNA fragments or the bulkier DNA fragment will not be able to travel much in a agarose gel, whereas the smaller DNA fragments, they will be able to travel much in these agarose gel, right. So the band which is far from those places where I have loaded my DNA of interest, that is of a lighter fragment, or that is having a less number of nucleotide as compared to those which is near to the wells where I have loaded the DNA fragments. So this is a gel electrophoresis which you have to explain. Now next is a selectable marker in PBR 322. PBR 322 is a vector. is a vector which carries our DNA or which carries our DNA of our interest, which is also called as a vehicle. So, in case of the PBR 322, two markers are present. These markers are resistant towards various antibiotics. One is called as a ampicillin resistance, and second one is called as a tetracycline resistance. Right. To talk about both of these, they provide resistance towards different antibiotics. When I insert a gene of interest near or in between this ampicillin resistance gene, the transformed cells, the recombinant cell will not be able to provide resistance against ampicillin. If I grow the cells in the ampicillin medium, the cells which are recombinant, they will not be able to grow. Whereas those cells which are now they will become a tetracycline resistant. If I grow them on tetracycline, they will be able to flourish at a very fast rate. So these are the two type of genes which is present in them. To talk about the vectors and the placement of these different resistance gene and ORI sequences and various restriction sites, they are as, this is one sequence which is called as a ORI, which is also called as a origin of replication initiation. This is called as a, this region they provide a ampicillin resistance, this region provides a tetracycline resistance. This provides a BAM-H1, which is also a restriction endonuclease site. This site is called as a PVU2. This is a SAL1. This is another PVU1. This is a CLA1. Sort of the different restriction enzyme sites are there. So, if a gene of interest is inserted into ampicillin resistance, the cell will not be able to show a resistance against the ampicillin if it try to grow on the ampicillin medium, but they will be able to show the tetracycline resistance, right. So, these are called as a selectable marker. Now, this was a question number 7. Let us move towards the next question. Draw a labeled sketch of sparged stirred tank. I am just making a rough diagram because in the session we have discussed about these things in detail. Whole diagram you have to draw. Even in this case, culture medium is present all over the place and they are having bubbles in it, and this sparged tank, sparged stirred tank, bioreactor, they move like this in a one direction like this, they keeps on moving, and this is a culture medium. These are the bubbles. This is called as a stirrer. First of all, tell me what is the significance of these bioreactors? Significance of bioreactor is that they help in culturing a particular cell, so the host cell. Suppose I want to make a copies of insulin hormone. That is produced with the help of a genetic engineering method, where a gene of interest that can be inserted into the cells, and those cells, they can be a 1 or 2 or the 1,000 in number, but I want a bulk of these products, they should be formed. This is produced in the bioreactor. From a one cell, the 2 copies, the 3 copies, in large number of the copies they will be produced. This is called as a bioreactor. For the preparation of the drug, for the preparation of the foods which are rich in proteins, the carbohydrate, different things, they are produced in the bioreactor only. So, this is our main use of this bioreactor. So, well labeled diagram, what is the use of this diagram, what is the function of this bioreactor, what are the application of this, that you have to write separately. Rest, let's head towards the next question. Question number 9. This is a 5-mark question. Unless the vector in the source DNA are cut, fragments separated and joined the desired recombinant vectors, that molecule cannot be treated. How are the desirable DNA sequence cut? That means we want that my DNA has to be cut into the various fragments, and we know the enzyme, and we have studied in this session also, that is a restriction endonucleases. Restriction endonucleases. Restriction endonucleases, they are also called as a molecular seizure, which cut at specific sequence. They need a palindromic sequences, they produce either a blunt end or the sticky ends. Same type of restriction enzyme will be used in the host DNA also, so that the same end that should be produced, a complementary end should be produced, because they read a specific sequence called as a palindromic sequences. Explain the technique used to separate the cut fragment. We all know that is a gel electrophoresis. How are the resultant fragments joined to the vector DNA molecule? Definitely they will require some enzyme, and in this case the enzyme which is required is called as a ligase. So this is not enough for a 5 marks, you have to explain about these procedure. Regarding the ligase, we have discussed about the ligase. What they do, they used to make a phosphodiester bond. They used to join the sugar phosphate linkage. Phosphodiester bond is formed, and you have to explain about each of these in detail. Let's talk about next question, the last question of today's class, that is related to very important topic, that is a PCR. Describe the different steps in a one complete cycle of the PCR. PCR is what? A polymerase chain reaction. In this, the different steps are there. First of all, the DNA which has to be amplified. Mainly the PCR function is for amplification. In case of the DNA, first you have to explain about denaturation, where the two fragments of the DNA, they are separated. They require a high temperature, around 95-degree temperature is required. After that is called as an annealing process. In the annealing process, the RNA primers, they are allowed to join. Because the DNA polymerase cannot read, they need a basically they need a free OH group, right? Then only they will extend that chain. For that, we always require primers, and that primer is joined with the help of that particular process is called as a annealing. So primers are used, RNA primers are used, and third one is called as a extension process. So in case of the extension, the DNA polymerase which is used, that is called as a Taq polymerase. We have explained about, we have already studied about the Taq polymerase, and who discovered the whole procedure? The Kary-Mullis. State the purpose of an amplified DNA sequence. So many purposes are there. Suppose in a crime scene I found the DNA is present and I want to amplify it. That is done with the help of a PCR. Normally what I want, at a particular gene is there, a DNA if interest is there, that give rise to a sequence. Suppose they give rise to insulin hormone. I want that should be amplified so that that can be inserted into a multiple host. That is done with the help of a PCR. So these are the different uses of the PCR. So we have done today around 10 questions from different category. Hope the things are clear to you. Hope you enjoyed the session also. We'll meet in the next session. We'll discuss about a new chapter. Till then, take care of yourself. Thank you so much, students, for watching this.